Good morning, everybody. I am Dr. Walvekar from Biochemistry Department. In the continuation of the classes on carbohydrate metabolism, in the last classes, we discussed about the pathways like glycolysis and then continued to the citric acid cycle, wherein there will be the 8 reactions will be taking place, whereas glycolysis is consisting of the 10 steps of the reactions which can take place both in aerobic and anaerobic conditions, whereas citric acid is the one which will be in the aerobic only, and the product that will be formed from the glycolysis pyruvate that will yield into the ester CoA, which will leading into the citric acid cycle, which is also called as the TCA cycle because of the formation of citric acid, tricarboxylic acid. It has been named by that one name also, and also as the one scientist called Krebs, he was the one who made the most of the contribution for this pathway to be found out, so that his name is also called many times. And citric acid cycle is the one which is referred to as the hub of the all the metabolic reactions, because it is the one which behaves like something like a circle in a city where all different roads come and meet together and get circulated. That way, the products of metabolic products of carbohydrate, fats, and proteins, they get come and attached to this one and get yielded into the formation of the fuel, that is the energetic products. So we'll just see that one citric acid cycle, how these reactions takes place, and we will see that one what is the importance of this citric acid cycle to the human body. Okay, it is considered as the central hub, as it is one which acts as the connecting one for the other different metabolic activities of fatty acids, amino acids, and the carbohydrates. And it is the one which brings about the catabolism of ester CoA, and there will be the end product like carbon dioxide and water molecules will be formed, along with the generation of the energy, that is in the form of the ATP molecules. So this is the circle, how it exactly takes place. So it is called as the circular one because the at the end of the eighth product, or the product that will be formed oxaloacetate, that will, again it moves into the metabolic reactions along with the another one ester CoA and continues the reaction. So that's why it is referred to be as the circular reactions that takes place. So we go by one the reactions that they take place. It is the one which takes place in the mitochondria, wherein it brings about catabolism of ester CoA to CO2 and H2O. In the step 1, there will be the formation of citrate will be there. This is the formation of citrate from the combination of oxaloacetate and ester CoA in the presence of citrate synthase. It is the enzyme. And once that oxaloacetate is joined with the ester CoA, a water molecule attached with the acetyl, leading to the release of coenzyme A from the complex. You can see the reaction here. So isoanoacetate, that is the end product of the reaction in the circle. It moves with what? The acetyl COA. In the presence of citrate synthesis, it forms into the citrate. It forms into the citrate. Second reaction, the citrate is rearranged just in the isomeric form by the enzyme called as aconitase. This aconitase reaction is the one which acts as a dual factor or a dual role in the enzyme and regulates the synthesis of the protein also, and also it regulates or controls the level of iron in the body. This is the role of aconitase, which converts the citrate into the isomeric form, that is the isocitrate will be formed. In these reactions, the water molecule will be removed and put back into the another location, that is from third to fourth position. How it is, just see, this is the water molecule will be removed from here, and here you can see the change here, that it makes the isomeric form of the citrate, that is isocitrate. Okay, and in the third step, that is the oxidation of the isocitrate takes place, and that will be converted into alpha ketoglutarate. There will be one generation of NADH. NADH will be formed at this reaction step, that is third step, where the isocitrate will be converted into the alpha ketoglutarate, and there will be the generation removal of the carbon dioxide molecule. Then there will be the formation of alpha ketoglutarate. Here the carbon dioxide molecule will be removed, and in the presence of isocitrate dehydrogenase, this isocitrate is getting converted into alpha ketoglutarate, and at the same time there will be a formation of or generation of one NADH molecule from the NAD. And reaction number 4 it is, that is alpha ketoglutarate. It is oxidized, it will be getting oxidized, and the carbon dioxide will be again removed, and coenzyme will be added, and there will be the formation of the product called as the succinyl CoA. Succinyl CoA is the one product that will be formed from the oxidation of the alpha ketoglutarate. Again here there will be the oxidation of NAD, which will be reduced to NADH. So we have to remember the step by step these reactions takes place. Here alpha ketoglutarate, it gets converted into the succinyl CoA in presence of one enzyme, that is alpha ketoglutarate dehydrogenase, and there will be reduction of NAD to NADH with the removal of one carbon dioxide molecule. So, in the reaction step number 5, the succinyl CoA, it will be converted into succinate. The energy that will be released, it will be used to make the GTP, that is guanosine triphosphate, from the GDP, and one inorganic phosphorus. It helps for the substrate level phosphorylation, and then there will be the one ATP will be made available by the GTP. So the enzyme which one is helping for the conversion of succinyl CoA to succinate is the succinyl CoA. This catalyzes the reaction, and here the GTP it promotes by providing or donating one phosphate molecule and from the phosphoryl group, and it is the generation of ATP at this substrate level. See, the succinyl CoA, this will be getting converted into the succinate, and the GTP is giving rise to the formation of one GDP, and this phosphorylation will be taking place by the donation of this phosphate molecule, and there will be the formation of one ATP molecule. In the step number 6, it is succinate, it gets oxidized to fumarate. Succinate is oxidized to fumarate. So here what happens during the course of oxidation, the FAD will be converted or reduced to the FADH2, and the enzyme that catalyzes this reaction is the succinate dehydrogenase. This will be catalyzing this reaction. The oxidation of FAD to FADH will take place, and there will be conversion of succinate to fumarate. Succinate to fumarate and FAD to FADH2. Step number 7, it is the hydration of fumarate to malate. This is the one where there will be the reversible reaction takes place. That is, the fumarase continues to rearrangement of the process by adding the hydrogen oxygen back into the substrate that has been previously removed one. So addition of the water molecules takes place in the mitochondrial membrane and generates to their continuity of the citric acid cycle. So there is the need that there should be the formation of malate, otherwise it will be impermeable for the oxaloacetate to go move inside. Later on the steps I will inform you. So the formation of the fumarate is important step here, that is hydration of the fumarate to malate. And then malate is oxidized to produce the oxaloacetate, that is the initial starting compound what it was used in the first step, where there is oxaloacid, it combines with the pyruvate in the presence of citrate, it will be forming into the citrate. So this is the how the 8-step reactions takes place, and while formation of this oxaloacetate from the malate, there will be the oxidation of the NAD takes place, and it will be reduced to the NADH. The oxaloacetate that is generated, it will combine with the another molecule of the acetyl CoA and continues the cycle, as I said earlier. So just if you see this, from malate to oxaloacetate it will be formed. Just when you see this reaction, you will come to know that oxaloacetate, it combines with the acetyl CoA with the presence of citrate synthase and forms into the citrate, and this one will be converted into the isocitrate in the presence of enzyme called as aconitase, and then isocitrate into the oxalosuccinate, and from oxalosuccinate to alpha ketoglutarate, and then to succinyl CoA, where there will be the formation of GTP will be there, and then succinyl CoA2 to succinate, and succinate to fumarate, fumarate to mannate, and then to the oxaloacetate, the final product. And this, during this course of reactions, what happens, there will be release of the ATP molecule. So this continuous reactions will be taking place with the help of every oxaloacetate end product of the one cycle that will be forming as a starting compound for the next cycle of the TCA cycle. Now, regeneration of the oxaloacetate. The TCA cycle, basically it involves the oxidation of the acetyl CoA to carbon dioxide with the simultaneous regeneration of the oxaloacetate, and means at the end of the reaction there will be the regeneration of the oxaloacetate, and there may never be the full complete consumption of the oxaloacetate or any other intermediate in the cycle. And another thing, the importance of this one is oxaloacetate. It is required to start the cycle. As you seen that one, it is the one works as the starting compound, and then very little amount of this one is required, and at the end of the cycle, it is regenerated by the oxidation of the mannate by the mannate dehydrogenase, so that oxaloacetate will act catalytically to restart the cycle. And in case there is the absence of this oxaloacetate, the TCA cycle will not operate in a complete manner, but on the contrary, it will result in the accumulation of acetyl CoA, leading to the formation of the ketone bodies. So if there is more amount of the ketone bodies will be there, so that leads to the decrease in the pH level, acidic condition will be there, and this can be more found with the diabetic patients. And another thing is that there is the requirement of the oxygen in this one. That is, there is no, though there is no direct involvement of the oxygen in this TCA cycle, this cycle completely operates in the aerobic condition. So you may remember that glycolysis is the one metabolic pathway which works or takes place in the presence of oxygen and also in the absence of the oxygen. Whereas this TCS cycle, it works under the aerobic condition. This is because NAD and the FAD, it required for the operation of the cycle. It can be generated from the respiratory chain only in the presence of the oxygen. Hence, it is considered that, or it is said that the TCS cycle, it works under only the aerobic condition. Now, at the end of this one cycle of the citric acid, one can judge that how many amount of the citric ATPs are formed in a cycle. So, in this oxidation of acetyl CoA to water molecule and carbon dioxide, there will be some generation or release of the fuel molecules in the form of the energy. Those things are this way, that 3 molecules of NADH and FAGL2 are produced for each molecule of the CA for one turn of the cycle. So when 3 molecules of NADH releases, so it leads to formation of 7.5 ATP. Whereas when FADH is formed, it generates 1.5 ATP molecule, and one of the one molecule of the ATP molecule is generated at the substrate level during the conversion of succinyl CoA to succinate. So what happens when after the addition of all these amount of the ATP that are formed, it amounts to the generation of 10 ATP molecules for the each turn of the one TCA cycle. So remember that this one works out in the presence of the oxygen, and the oxidation of the acetyl CoA, it releases the water molecule and carbon dioxide. Along with return, the fuel energy molecules will be released, and these are at the end of the cycle there will be release of 10 ATP molecules at the each turn of the TCA cycle. So what is the significance of the TCA? It is there will be the complete oxidation of the acetyl CoA takes place. This is the one which will be formed from the breakdown of the three macromolecules, that is the carbohydrate, fats and proteins, and there will be the generation of ATP molecules in the form of the energy. And another thing is that it is a final common oxidative pathway. TCA cycle is the one which is considered as the final common oxidative pathway, and it is the one which makes or it shows the integration of the pathway, and fat will be burned at the peak of the carbohydrate, means at the expenditure of the carbohydrate, fat will be utilized and formed into the fuel one, and excess of the carbohydrates, whatever there will be there, first thing it will be stored as the glycogen, and if there is more amount of the carbohydrates, that will be stored in the adipose tissue in the form of the neutral fat, and there won't be any synthesis of the carbohydrates from the fat. Actually it is from the carbohydrates to fat it will be. And another important thing is that the supplementation of carbon skeletons of the amino acid, finally they enter the TCA cycle. So these are the certain important things or significance of the TCA cycle. Another thing is that the TCA is called as the amphibolic in the nature, means it is the one which functions for the both the things, means the definition of the metabolism will be attained by this in the TCA cycle, that is it attains the function of catabolic and also the anabolic also. In the catabolic, what will be happening, that two carbon compound acetyl CoA produced from the metabolism of the carbohydrate, lipids and proteins, they get oxidized and they produce the ATP along with the carbon dioxide and water molecules. That is the one which is related to the catabolic role of this TCA cycle. Whereas when you consider the anabolic or synthetic role, the intermediates of the TCA cycle are utilized for the synthesis of the various compounds. In the circle of 8 reactions, there will be the formation of some intermediates which will be helpful for the synthesis of other compounds. That is the role of the anabolic one. First is the transamination. Transamination means the transferring of one amino group from the amino acid to the keto group, that will be considered in the metabolism of the proteins. That way you will learn about it very elaborately. This is the function that can be done, and this will be applicable for the synthesis of non-essential amino acids, and in that one the pyruvate will be converted into the alanine, That is the transfer of the amino group to the keto acid from the amino acid. Similarly, the oxaloacetate, it gives rise to aspartate, and alpha ketoglutarate, that is the intermediate product which gives rise to the formation of the glutamate. Like this, certain amino acids which you can label them as the non-essential amino acid. The non-essential because they can be synthesized in the body. So these can be done anabolically in the under the heading of this function or the process called as the transamination. This can be attained by this TCA cycle. One is that is the formation of the glucose, that is gluconeogenesis. Means when there is no availability of the glucose, then the formation of glucose will be done by the non-carbohydrate sources like alanine, glutamine. They contribute to this process called as the gluconeogenesis, where there will be the carbon skeletons of these things when they enter into the TCA cycle after the attending the functions like deamination. Deamination means removal of the amino acid, whereas transamination means it is the transfer of the amino acid to the keto acid. So, and after attending these two functions, the carbon skeletons of these amino acids can be transferred through the TCA cycle for the production of the glucose through the method called as the or the step called as the gluconeogenesis. And third one is the fatty acid synthesis. Acetyl CoA formed from the pyruvate, it is the starting material for the long chain fatty acid. That is, palmitic acid can be formed from the acetyl CoA. So these are the functions which are attained to the anabolic functions of the TCA cycle. And then there can be the formation of cholesterol, that is the starting material, that is acetyl CoA, that will be used for the synthesis of cholesterol, which is required again for the synthesis of the steroids. That is not only this one, but also this formed cholesterol can be one which is utilized for the formation of vitamin D also. And fifth function of this TCA cycle is that is the succinylcycline CoA that is formed in this TCA cycle as an intermediate one. It is taking part in the heme synthesis. So you can come to know about it in detail when you learn about the heme synthesis or the heme metabolism. Okay. So this is how the TCA cycle it is getting involved into the both the type of the reactions, that is the catabolic and anabolic. Hence this one is called as one which is attending both the dual part. That is why it is called as the amphibolic reaction step. Now citric acid, it gets regulated at certain positions where the reactions will be reversible one. So in the next slide we will go there. See, it is the first step where this, if the more amount of the ATP is formed, it stops the reaction so that it will be checked there only. And second one is at this enzyme level, that is isocitrate dehydrogenase. Here also the checking of this one will be done to regulate the citric acid cycle. And third one is the alpha ketoglutarate, where there will be the regulation will be done. And finally pyruvate dehydrogenase also. that I will show in the next slides. First one, that is, this regulation of the citric acid is very much vital one, because if it is not checked, then it may lead to the accumulation of the more amount of the energy, or large amount of the energy will be wasted also. So to the maintaining this wastage or curbing the wastage of the metabolic energy, it should be regulated, and it will be regulated at the different steps. The reactions which are involved in that one are considering of three enzymatic levels. That is, one is the citrate synthase, isocitrate dehydrogenase, and third one is the alpha ketoglutarate dehydrogenase. These are the one which are responsive for the formation or regulation of this fuel molecule that will be formed here, and which will be represented by the ATP by ADP and NAJ to NAD ratios. So because of these regulatory actions, the citric acid cycle can be regulated. So first one is the citrate synthase. It is responsible for the rate of reaction in the first step, and it will, what it will do, when acetyl CoA is combined with the oxaloacetate to form the citrate, it inhibits by the high concentration of the ATP, acetyl CoA and NADH, which indicates already the presence of high amount of the energy. And the molecule that is present in the reaction citrate, it can itself can act as an inhibitor of the reaction. So at the level of this action of this citrate synthase, the reaction, it gets regulated by indicating the development or the formation of high concentration of the ATP, so that the further reaction steps will be inhibited. The second one is the isocitrate dehydrogenase. It regulates the speed at which citrate isomer isocitrate, it loses the carbon to form the 5 carbon molecule, that is alpha ketoglutarate. Here the NADH is the product of the reaction, and when the more amount of this NADH is formed, then the isocitrate dehydrogenase, it acts as the inhibitor and it displaces the NAD molecules, means it stops the reaction there only. And third one is the alpha ketoglutarate dehydrogenase. This also curbs the reaction when there is the more amount of formation of the succinyl CoA and NADH, and it inhibits the reaction when there is more amount of this NADH is formed. So there are three enzyme levels are there. One is citrate synthase, second one is isocitrate dehydrogenase, third one is alpha ketoglutarate dehydrogenase, and another one, that is pyruvate dehydrogenase. What happens? Certain tissue or the organs like the brain, it is totally dependent upon the supply of the glucose only for the sake of getting the supply of acetyl CoA. So in that one, what happens? The action of this pyruvate dehydrogenase will be inhibited by the more amount of formation of the ATP, and again the regulation will be taking place at the brain level. Next one is, that is, the calcium will also act as a regulator for the citric acid cycle. It is an important regulator in the cycle, and increase in the both ADP and calcium are the consequence. Changes in the activity will be brought down, and calcium ions, they activate isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase at the third and fourth steps of the cycle. So the calcium is also involved in the regulation of the citric acid cycle. And one can ask you in the questions or in the viva, just you mentioned about the involvement of water soluble vitamins or B complex vitamins which are involved there as the one in the form of coenzymes for the various carbohydrate metabolism. That time you have to answer that the one Lysine, it is that in the form of the NAD and will be involving in these reactions which converts itself by NAD to NADH, and another enzyme that is the TPP, that is thymine pyrophosphate from the thymine. It is the one which gets involved in the reaction with the enzyme involved in the taking place in the alpha ketoglutarate dehydrogenase, and riboflavin, that is as the flavin adenine dinucleotide reactions. That time it is riboflavin is the one which in the form of FAD it takes part in the reactions related to the succinate dehydrogenase, and pantothenic acid, it acts as a coenzyme A. Like this, the many coenzymes which are considered from having the root level observation are their sources in the water soluble vitamins or the B complex vitamins. They can be labeled as that one which are required for the various this carbohydrate metabolism. And coming to this inhibitors of the citric acid, that is one fluoroacetate. It will be used as a rodenticide, that is actually it will be used for the rat killing. It inhibits the activity of the aconitase, that is the one enzyme which is used at the first step. So that will converted into fluoroacetate CoA and gives rise to fluorocitrate after condensing with the oxaloacetate. So the aconitase is inhibited by the fluoroacetate. This will be the one which acts as the inhibitor for the action of these enzymes. And second one, alpha ketoglutarate dehydrogenase, it is inhibited by the some arsenic compounds. And malonate, it is a competitive inhibitor for the succinate dehydrogenase. So these are the some three compounds which will be acting as the inhibitors for the citric acid cycle. So we start with just the introduction to the gluconeogenesis. So what is the gluconeogenesis? As I said in the various earlier 2-3 classes, it is the one, the metabolic step wherein there will be the formation of the glucose from the non-carbohydrate compounds or from the glycogen, because the glucose supplementation is very much mandatory. It is synthesized from the pyruvate, which is derived from the glucogenic amino acids, intermediates of the TCA and glycerol. And gluconeogenesis is the one which consumes the energy. You should know that the difference between the gluconeogenesis and the glycolysis or the Krebs cycle. Whereas in the Krebs cycle and the glycolysis, there is the catabolism is taking place, and what happens, there will be the release of the energy, that is in the form of the fuel, or you can say that the ATPs will be formed. Whereas here in this process there will be the consumption of the energy will be there. So the need of this gluconeogenesis in the body, why it is needed? Because some of the activities they need absolute supply of the glucose to meet the need of these some certain organs like the brain one I can tell, or erythrocytes, testes and the kidney. Human brain alone requires about 120 gram of the glucose per day, and it is required for the maintaining levels of the intermediate of the TCS cycle. It serves as the fuel for the skeletal muscle in the anaerobic conditions. So for all these requirement, there is the need of the glucose. So body has made the arrangement of supplying of the glucose from the other sources in the body, which one will be referred as the gluconeogenesis. So it is the mechanism which is required to clear the products of other tissues from the blood. That is, glycerol which is continuously produced by the adipose tissue by the action of one enzyme called lipolysis of the TG. And during the starvation, gluconeogenesis maintains the blood glucose level. So when there is no supply and if the person is under the starving condition, then the stored glycogen will be depleted for 12 to 18 hours and it converts that into the glucose and makes the supply of the glucose to the body. However, during the course of the longer period or prolonged starvation, it is speeded up by the method, means apart from the source as the glycogen, it will put up to the protein catabolism and it will be taking the help of glycogenic amino acid. Breakup of this one will be taken place and glucose will be formed and it will be supplied to the these what mentioned the parts are the erythrocytes or the brain. It will be supplied through with the glucose only. Gluconeogenesis, it occurs mainly in the liver and also in the kidney or the renal cortex, and these organs they will be possessing the some enzymes that are required for the gluconeogenesis, and this step it takes place in the mitochondria partly and also partly in the cytoplasmic, and about 1 kg of the glucose is synthesized every day. So regarding the reaction of gluconeogenesis we will take in the next class. So we have seen that one there are 8 steps of reactions in the citric acid one, where there will be at the end of the reaction where the final product, that is oxaloacetate, is formed. That one is getting involved, or the generated oxaloacetate takes part in the fresh first step reaction with the another molecule of the TCA cycle with the acetyl CoA, and during that 8 steps of the reactions there will be release of finally the 10 ATPs will be formed, and also we have seen some of the regulations which will be regulating the Krebs cycle or TCA cycle you say. So that will be done at the 3 stages. Finally, if there is the more amount of the ATP is formed, the further reactions will be stopped there or it will be inhibited. And we have seen also the regarding the course of gluconeogenesis, which is taking place when there is no supply of the glucose from the carbohydrate. The body will make arrangement for the continuous supply of the carbohydrate from the other sources. So these steps of the reactions is called as, or the pathway is called as the gluconeogenesis. And first it will take the help of, or it will look at the sources of the fat reservoir. Later on, in the longer starvation condition, when there is no supply of this carbohydrate or the glucose, it will take the help of the protein molecules, and somehow it makes the arrangement, or it takes the conditions that the glucose supplementation will be there always to the very important vital organs. So the reactions that are involved in this gluconeogenesis, we will deal it in the next class. Okay, thank you.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.